Picture yourself standing on the deck of a research vessel somewhere off the coast of Namibia. The year is 2004. The water around the hull has changed color. It is no longer blue, it is greenish-white, spreading in every direction for tens of kilometers, and the smell reaches you long before the visual does. Rotten eggs, concentrated. The kind of concentration that makes your eyes stream and your throat close. Dead fish are surfacing by the hundreds of thousands. The crew recognizes this. It has happened before. A mass of hydrogen sulfide, a gas lethal to most aerobic life, has erupted from the seafloor and is now venting through the water column and into the open air. This is not a historical event. This happened in the modern era. And the chemistry driving it is identical to the chemistry that dominated Earth's oceans for over 1 billion years. Before we go any further into what that chemistry means and where else it is currently running, subscribe to this channel. What you are about to hear is not speculation. Every mechanism on this list has a geological record. Everyone has a modern parallel already in progress. What the researchers documenting these patterns are measuring is not a prediction. It is a recurrence. Number 1. The Canfield Ocean. The chemistry that poisoned 1 billion years is waking up. In 1998, geochemist Donald Canfield of what is now the University of Southern Denmark published a paper in Nature that restructured the accepted history of Earth's oceans. For decades, the Standard Model described the ancient oceans as occupying one of 2 states, oxygenated or anoxic, depleted of oxygen but otherwise chemically inert. Canfield proposed a 3rd state, one where oxygen was absent and sulfate-reducing bacteria had converted the water column into a solution saturated with dissolved hydrogen sulfide, simultaneously oxygen-depleted and sulfur-saturated. The technical term is euxinic, derived from Pontus Euxinus, the ancient Greek name for the Black Sea. What Canfield proposed was not an aberration, it was a baseline. The Canfield Ocean describes conditions that prevailed across most of Earth's deep and intermediate ocean for approximately 1.2 billion years, from roughly 1.8 billion to 540 million years ago. That interval is sometimes called the boring billion. The description is a geological misnomer. The chemistry during that period was not inert. It was actively hostile to nearly all complex life. The biological engine driving it was a class of microorganisms called purple sulfur bacteria, genus Chromatium, which performed photosynthesis using hydrogen sulfide as an electron donor rather than water. Their blooms were vast enough to give the ocean a deep violet coloration visible from altitude. The byproduct of their metabolism, hydrogen sulfide gas, vented continuously into the atmosphere, reacting with other compounds to produce a permanent pale green haze across the sky. The evidence for the Canfield Ocean is geochemical. Iron speciation analysis of ancient sedimentary rock reveals the signature of sulfide-rich water. Dissolved iron was stripped from the water column and precipitated as pyrite, fool's gold, on the seafloor. Entire ancient ocean floors became iron graveyards. Biomarker analysis, specifically the detection of ixorinierotene, a pigment unique to green sulfur bacteria, confirmed that euxinic conditions extended into the photic zone, the sunlit upper layer of the ocean, rather than remaining confined to the deep. Photic zone euxinia is the technical term for sulfidic conditions reaching the surface. Its specific horror is structural. Even shallow coastal water becomes lethal to aerobic organisms. There is no refuge in the shallows. There is no escape to the surface. The chemistry requires 2 conditions: low atmospheric oxygen and high sulfate concentration. When oxygen is insufficient to decompose organic matter on the seafloor, that matter fuels sulfate-reducing bacteria. Those bacteria produce hydrogen sulfide as a waste product. A hydrogen sulfide builds in the water column. Once it reaches the photic zone, photosynthetic sulfur bacteria colonize it. They produce more sulfide. The system becomes self-reinforcing, a closed feedback loop that, once established, resists disruption for geological timescales. The mainstream interpretation holds that the Canfield Ocean was a stable thermodynamic attractor, not a catastrophe, but a default state that Earth's chemistry fell into when atmospheric oxygen was insufficient to prevent it, and that the Great Oxidation Event gradually destabilized it. Some researchers argue that euxinic conditions were patchy and basin-restricted rather than globally uniform. pointing to isotopic heterogeneity in the rock record. The most speculative reading, discussed among astrobiologists, is that purple sulfur bacteria may represent the default photosynthetic life form for ocean-bearing planets across the universe. That Earth's current green biosphere is the anomaly, not the baseline. The Black Sea today is euxinic below approximately 150 meters. The Baltic Sea generates expanding sulfidic dead zones annually. A 2019 paper by Schieber and colleagues documented isoriniaritane biomarkers, the identical chemical signature of ancient photic zone euxinia, forming in modern restricted basins. The chemistry has not been retired. It is dormant in the baseline, waiting for the oxygen budget to shift far enough. But the deep water euxinia documented by Canfield has a coastal counterpart, one that erupts without warning, covers tens of thousands of square kilometers in hours, and is already accelerating. Number 2, the shelf eruptions. The gas that defined the killing chemistry of the ancient ocean is already surfacing on modern coastlines. The Benguela upwelling system off the coast of Namibia and South Africa is among the most biologically productive ocean zones on Earth. Cold, nutrient-rich water rises from depth, fueling explosive plankton growth. When that planktonic biomass dies and sinks, decomposition bacteria consume dissolved oxygen across the shelf's seafloor. The result is a layer of anoxic, organic-rich sediment, sometimes called the mud belt, extending across hundreds of kilometers at depths between 80 and 300 meters. Within this sediment, sulfate-reducing bacteria produce hydrogen sulfide continuously. Under normal conditions, the gas dissolves into sediment pore water or reacts with iron minerals before reaching the water column. But when pressure differentials, storm disturbances, or temperature shifts destabilize the sediment, the gas erupts. In 2004, an eruption event released enough hydrogen sulfide from the Benguela Shelf to discolor roughly 20,000 square kilometers of surface water greenish-white, the visible signature of elemental sulfur particles suspended in the water column. Dead rock lobsters, fish, and benthic invertebrates carpeted beaches along the Namibian coast for weeks. The smell was described by coastal residents as an invisible wall. Oceanographic sampling documented H2S concentrations in the water column reaching levels lethal to most marine animals within minutes of exposure. That event was not unprecedented. Similar eruptions had been documented in 1994 and 2000. They recurred in 2009 and subsequent years. Satellite imaging, capable of detecting the sulfur discoloration patterns from orbit, has documented a measurable increase in both the frequency and spatial extent of these events over the past 2 decades. Hydrogen sulfide at 50 to 100 parts per million destroys the olfactory system of vertebrates on contact. At 1,000 parts per million, neurological collapse is near instantaneous. It is denser than air, which means it accumulates in low-lying coastal areas and enclosed bays when surface venting events are large enough. During major Benguela eruptions, the chemistry at the water surface is not metaphorically reminiscent of ancient euxinia. It is chemically identical to it. The geological precedent is direct. Lee Kump of Pennsylvania State University published a 2005 paper in Nature proposing that hydrogen sulfide erupting from euxinic oceans into the atmosphere contributed directly to the terrestrial extinction of the end Permian event, 252 million years ago, degrading the ozone layer and poisoning land ecosystems from the coastline inward. The Namibian shelf events operate the same mechanism scaled down by approximately 5 orders of magnitude. The mainstream interpretation holds that Benguela eruptions are a natural feature of highly productive upwelling systems, intensified by eutrophication from agricultural runoff, but not yet critically different from pre-industrial baselines. Some researchers argue the eruptions are measurably increasing in both frequency and spatial extent, all correlating with rising bottom water temperatures and expanding oxygen minimum zones, and that the trajectory is directional rather than oscillating. The most speculative view is that the Namibian shelf represents a prototype, the first large coastal system beginning its transition toward persistent shelf euxinia, and that similar transitions may be imminent in the Humboldt upwelling system off Peru and Chile, one of the most productive zones in the global ocean. What is not speculative is the escalation in the measurement record. The chemistry is not hypothetical. The eruptions are documented, they are increasing. And directly beneath the seafloor sediments driving these events, a far larger reservoir of gas exists. One that, if destabilized, would not discolor a coastal bay. It would alter the atmosphere of the entire planet. Number 3. The clathrate gun. The frozen methane beneath the ocean floor has fired before. The trigger is being pulled again. Approximately 56 million years ago, at the boundary between the Paleocene and Eocene epochs, something injected an enormous quantity of isotopically light carbon into the atmosphere and ocean in a matter of centuries, possibly decades. Global temperatures rose by 5 to 8 degrees Celsius within approximately 20,000 years, a geological eye blink. The event is called the Paleocene-Eocene Thermal Maximum, the PETM. Its cause remains debated. Its chemical signature is not. James Zakos of the University of California. Santa Cruz and colleagues published the foundational PETM analysis in Science in 2001, documenting the carbon isotope excursion preserved in deep-sea sediment cores, a global negative shift in the ratio of carbon-13 to carbon-12, consistent with carbon sourced from biological or organic material, not volcanic rock. Gerald Dickens of Rice University proposed in a 1995 paper in geology that methane clathrates, ice-like structures in which methane molecules are trapped within water lattices at specific temperature and pressure conditions along continental margins, destabilized in a cascade, releasing hundreds of gigatons of methane into the water column and atmosphere within centuries. Methane hydrate deposits exist primarily at depths between 300 and 500 meters on continental shelves worldwide. The estimated global inventory ranges from 500 to 2,500 gigatons of carbon. The lower bound of that estimate represents more than twice the carbon currently in Earth's entire atmosphere. Methane is approximately 25 to 30 times more effective at trapping atmospheric heat than carbon dioxide over a 100-year time scale. When a warming event raises deep ocean temperatures past the stability threshold for clathrates, they decompose. Methane rises. It warms the atmosphere. The atmosphere warms the ocean. More clathrates decompose. This is the clathrate gun hypothesis, a self-amplifying release cascade with no natural break once initiated above a critical threshold. During the PETM, ocean pH dropped sharply, dissolving carbonate sediments across the seafloor in a distinct horizon visible in cores as a rust-red clay layer, technically described as carbonate compensation depth shoaling. Benthic foraminifera, or single-celled organisms that build calcium carbonate shells, suffered a 30 to 50% extinction rate. They did not die from heat or predation, they dissolved. The mainstream interpretation holds that the PETM was likely triggered by volcanic activity associated with the North Atlantic igneous province, and that clathrate destabilization amplified the warming, but was probably not the sole primary driver. Dickens and subsequent researchers argue that the isotope excursion is too large and too rapid to be explained by volcanism alone, and that large-scale methane release must be invoked. The most speculative reading, held by a minority of climate scientists, is that Arctic continental shelf clathrates are already past their stability threshold in several locations, meaning the gun has not yet fired, but the trigger is depressing incrementally with each year of warming bottom water. A 2020 paper in Nature Communications, led by Orion Gustafsson of Stockholm University, documented active methane seep fields on the East Siberian Arctic shelf at densities not previously recorded in the measurement era. The PETM is the closest geological precedent for what the current carbon trajectory, if sustained, is capable of producing at ocean scale. The chemistry is the same. The sediment reservoirs are the same. The warming is the same in direction, if not yet in magnitude. But thermal stress on the ocean produces a second chemical consequence that operates alongside methane release, one that attacks the structural foundations of marine life from the molecular level up. Number 4. The acid ocean. 1. The torsion event dissolves shells from the inside. It is already repeating. 183 million years ago, during the early Jurassic, the Karoo Large Igneous Province erupted across what is now South Africa and Antarctica. Rising magma passed through buried coal and shale deposits, thermally decomposing them. The result was a sustained injection of carbon dioxide and methane into the atmosphere lasting hundreds of thousands of years. Ocean chemistry shifted so rapidly that organisms building calcium carbonate shells could not adapt. Their shells began to dissolve while the animals were still inside them. Stephen Hesselbo of the University of Exeter and colleagues published the key evidence in Nature in 2000. Directly linking the Karoo eruptions to the torsion oceanic anoxic event through a negative carbon isotope excursion in organic carbon from Yorkshire, England. Subsequent work by Micha Rule, also at Exeter, published in Science in 2011, tightened the chronology. The carbon injection likely occurred in under 35,000 years. In geological terms, that is instantaneous. pH reconstructions from boron isotope analysis of sediment cores, published in 2019, estimated the ocean surface pH dropped by approximately 0.3 to 0.4 units during the torsion event peak. Ocean pH today sits at approximately 8.1, down from approximately 8.2 at pre-industrial baselines. A drop of 0.1 units since the Industrial Revolution. That number seems small. pH is a logarithmic scale. A drop of 0.1 units represents approximately a 26% increase in hydrogen ion concentration. A drop of 0.3 to 0.4 units, as occurred during the torsion, represents a doubling to tripling of acidity. The distance between current ocean chemistry and Toarcian chemistry is a factor of 3, not an order of magnitude. The critical variable is carbonate saturation. As pH drops, the saturation state of aragonite and calcite, the minerals from which shells and coral skeletons are constructed, decreases. Below a threshold called the aragonite saturation horizon, structures built from aragonite dissolve faster than organisms can rebuild them. During the Toarcian, this horizon rose toward the surface in shallow tropical seas. Organisms could not extract sufficient calcium carbonate from the acidified water to build normal skeletal structures. The fossil record from this interval shows deformed shells, absent shells, and miniaturized body plans. Marine animals 50 to 80% smaller than their pre-eruption ancestors survived the aftermath. Metabolic stress and resource scarcity selected for smaller bodies. The mainstream interpretation, established by Hesselbo and Rule, holds that the torsion event represents a direct geochemical analog for modern ocean acidification driven by fossil fuel combustion, differing primarily in the rate of carbon injection. The torsion event unfolded over tens of thousands of years. Modern acidification is occurring over centuries. Some researchers argue the Toarcian analogy overstates the severity of modern acidification because the Toarcian event was compounded by simultaneous warming and anoxia that amplified its effects beyond acidification alone. The most speculative reading proposes that the torsion represents the minimum threshold model, the least severe acid event that still produced measurable extinctions, making it the lower bound of concern rather than the upper bound. Richard Feely, of the National Oceanic and Atmospheric Administration, published data in 2012 showing corrosive water already contacting pteropod shells along the United States Pacific coast in the modern era. Pteropods, small free-swimming mollusks that form the base of many polar food chains, are exhibiting shell dissolution in the Southern Ocean today. This is Toarcian chemistry. Present tense, not projected, not modeled, measured. The dissolution of shells, however, is only the surface expression of a deeper problem. The mechanisms that starve ocean water of oxygen, running simultaneously with acidification, produce a different kind of death, and they have been intensifying since 1960. Number 5. The Devonian dead zone. The eutrophication cascade that murdered an ocean is running again. 375 million years ago, the first forests developed root systems deep enough to chemically and physically grind bedrock into soil, phosphorus and nitrogen, locked inside continental rock for billions of years, dissolved into rivers and carried to the sea. The sea bloomed green from horizon to horizon in algal masses covering thousands of square miles, and then the oxygen vanished and the sea died. Thomas Algeo of the University of Cincinnati and Stephen Scheckler of Virginia. Tech published the foundational analysis in 1998 in Philosophical Transactions of the Royal Society, directly connecting the evolution of terrestrial plants to late Devonian marine extinction through nutrient runoff and eutrophication. The geological fingerprint is the Kellwasser black shales, sedimentary layers so oxygen depleted they are jet black, found at sites across North America, Europe, and Australia. The late Devonian extinction eliminated approximately 75% of all species over pulses spanning roughly 25 million years, with the most severe events at the Frasnian-Famennian boundary around 374 million years ago. Eutrophication operates as a cascade. Nutrient injection triggers algal bloom. Bloom biomass dies and sinks. As decomposition bacteria consume dissolved oxygen in the water column, oxygen drops below the threshold for aerobic life. The water column stratifies, an oxygenated surface layer above an anoxic, sulfide-rich deep. Over time, the sulfidic layer expands upward. Given sufficient time and nutrient input, the photic zone itself becomes euxinic. The entire column, from seafloor to surface, becomes hostile to aerobic life. This is the same mechanism operating in the Gulf of Mexico hypoxic zone every summer, driven by nitrogen and phosphorus runoff from agricultural fields in the Mississippi River Basin. The zone averages 15,000 to 20,000 square kilometers annually. In 2017, it reached 22,730 square kilometers, the largest measurement on record. Nancy Rabelais of Louisiana State University has documented the zone's chemical parameters and expansion over decades, and published its chemistry and science in 2002. The measurements match Devonian black shale formation conditions. Worldwide, there are now more than 700 documented coastal hypoxic zones, up from roughly 45 in 1960. The rate of increase was documented by Robert Diaz of the Virginia Institute of Marine Science and Rutger Rosenberg in a 2008 science paper. That rate mirrors the early stages of every documented eutrophication-driven extinction in the fossil record. The mainstream interpretation holds that the Devonian extinctions resulted from eutrophication compounded by global cooling driven by CO2 drawdown from expanding forests, a two-phase kill mechanism. Or some paleontologists argue volcanism and tectonic activity coincident with forest expansion contributed heavily, and that biological weathering alone cannot explain the full severity. The most significant observation is structural. The Devonian did not require a single catastrophic event. It required a threshold to be crossed by accumulating chemistry. The process currently underway is the same accumulation. The threshold is not fixed. It moves as ocean conditions shift. But some of what the ocean floor is accumulating today has been there far longer than any modern dead zone, longer in some cases than modern humans have existed. And it is beginning to thaw. Number 6, the frozen archive. Ancient viruses are being recovered from deep permafrost and ocean sediment, and some of them are still functional. In 2014, Jean-Michel Claverie and Chantal Abergel of the Structural and Genomic Information Laboratory at Aix-Marseille University published a paper in PNAS announcing the revival of a functional virus from 30,000-year-old Siberian permafrost. They named it Pithavirus sibericum. It measured 1.5 micrometers in length, larger than many bacteria, visible under a standard light microscope. It had been frozen since the late Pleistocene. Under laboratory conditions, it infected amoeba cells and began replicating within hours of thawing. In 2022, the same team revived Pandoravirus yedoma from permafrost dated to 48,500 years old, the oldest functional virus recovered from any source. The paper, published in Viruses, identified 13 distinct ancient viral lineages from 7 different Siberian permafrost samples, all including 5 new genera, with no close modern relatives. These are not variants of viruses currently circulating in global ecosystems. Their host ranges, replication mechanisms, and immune evasion strategies evolved under conditions that have not existed for tens of thousands of years. Modern organisms have no evolutionary history of exposure to them. The permafrost of Siberia, Alaska, and the Arctic continental shelves has been accumulating biological material since the Pleistocene. It preserves not only organic chemistry, but intact cellular structures, functional proteins, and, as Claverie and Aubergel demonstrated, viable infectious particles. Claverie has estimated that Arctic permafrost holds an average of approximately 10,000 viral particles per gram of soil, nay across a permafrost extent of approximately 25 million square kilometers. As permafrost thaws and releases its contents into rivers and ultimately into the Arctic Ocean, those particles enter aquatic ecosystems. Most will be non-functional, some fraction will not be. Marine sediment adds a second reservoir. Unlike permafrost, deep-sea sediments are not frozen, but in the anoxic cold conditions of the deep-sea floor, biological material degrades extraordinarily slowly. A 2016 study published in Proceedings of the Royal Society documented viral sequences in deep-sea sediment cores predating the Holocene. The specific risk from marine viral revival is not a cinematic outbreak scenario. It is subtler and more systemic. Marine microbes drive nitrogen cycling, carbon export, and oxygen production in the ocean. Novel viral pressures introduced into microbial communities can disrupt that structure. Disruption at the microbial level propagates upward through food chains in ways that are slow, difficult to detect, and extremely difficult to reverse. The ocean is not a collection of fish. It is primarily a microbial system. The fish are secondary. The mainstream interpretation in virology holds that the probability of ancient viral revival causing large-scale harm is low. Most ancient viruses targeted host species that no longer exist. Claverie himself, in a 2023 commentary in Nature Microbiology, argued that the unprecedented pace of permafrost thaw is releasing viral and microbial material faster than any previous interglacial event, creating conditions for which no geological precedent exists. The most speculative concern among a minority of biosecurity researchers is not the viruses themselves, but the ancient bacteria they carry, particularly those with novel antibiotic resistance profiles selected by a pre-antibiotic world. As of 2024, global permafrost is thawing at rates not observed in at least 11,700 years, according to data published by researchers at the Alfred Wegener Institute. The archive is opening. Its contents are largely unknown. What is known is that previous interglacials did not thaw permafrost at the current rate, and that the biological material accumulating across 25 million square kilometers of frozen ground represents a sample of every pathogen that circulated in northern ecosystems for the past several 100,000 years. Number 7. The Black Ocean Events. At least 6 times in Earth's history, all large volumes of the ocean lost their oxygen entirely. The preconditions for the next one are currently measurable. At specific depths in sediment cores drilled from the deep Atlantic and Pacific, the material turns black. Not dark, fully black. The color of seafloor sediment that has not contacted oxygen in more than 90 million years. These layers are called oceanic anoxic events. They are found in drill cores from every major ocean basin at the same depths, corresponding to the same geological moments. The chemistry that produced them was global and simultaneous. Hugh Jenkins of Oxford University has spent decades documenting the Mesozoic oceanic anoxic events, publishing analyses in the Annual Review of Earth and Planetary Sciences and multiple specialist volumes. At least 6 major OAEs occurred between 250 and 80 million years ago. OAE2, at the Cenomanian-Turonian boundary approximately 94 million years ago, eliminated approximately 8% of marine species. Black shale deposits from that event span an estimated 75% of the preserved ocean floor record. Sea surface temperatures in tropical regions exceeded 35 degrees Celsius. Deep ocean circulation effectively stopped. The triggers for individual OAEs varied. The Caribbean Large Igneous Province for OAE2, the Ontong Java Plateau, eruptions for OAE1 approximately 120 million years ago. In each case, volcanic CO2 drove warming, which reduced deep ocean oxygen solubility, which allowed oxygen minimum zones to expand until they merged across basins. The ocean stratified, circulation ceased, the deep became permanently anoxic. The physics is not complicated. Warm water holds less dissolved oxygen than cold water. This is a basic property of gas solubility. As ocean temperatures rise, the oxygen minimum zone, the natural mid-depth layer where dissolved oxygen is lowest, currently sitting between approximately 200 and 1,000 meters depth globally, expands both downward and upward. In a sufficiently warm and stratified ocean, it reaches the photic zone. At that point, aerobic respiration becomes impossible above the deep seafloor. Lisa Leven of Scripps Institution of Oceanography has documented measurable oxygen minimum zone expansion in the modern Pacific and Indian Oceans, across multiple publications. The volume of low oxygen water in the global ocean has increased by approximately 2% since the mid-20th century. That rate is accelerating, not stabilizing. The mainstream interpretation holds that Mesozoic OAEs required large igneous province volcanism to initiate, a forcing agent not currently active at equivalent scale, and that the warming and eutrophication driving modern oxygen loss are insufficient alone to produce a true OAE within any near-term time frame. Some researchers publishing through the Integrated Ocean Drilling Program have argued that the modern ocean's thermal stratification already mirrors pre-OAE states in several basins, and that a sufficiently large additional perturbation could trigger a transition that existing ocean chemistry would then sustain independently of the initial forcing. The most speculative reading is that OAEs are not exceptional events requiring extraordinary triggers, but the ocean's default condition under sustained high CO2 forcing, meaning the current trajectory does not need a volcanic trigger. It just needs time. The eastern tropical Pacific already contains a permanent oxygen minimum zone, where waters below 200 meters contain less than 2 millimoles of dissolved oxygen per cubic meter. OAE black shales formed by the same process. The difference is duration, not chemistry. And when all of these processes run simultaneously, the euxinia, the acidification, the anoxia, the methane release, they do not simply add, they multiply. The geological record contains one event where all five ran at once. Its chemistry is already detectable in modern ocean sediments. Number 8. The Great Dying's chemistry. The specific molecular signature of Earth's worst mass extinction is measurable in present-day ocean sediments. In 2005, Cliddy Grice of Curtin University in Australia and colleagues published a paper in Science that answered a forensic question 252 million years old. What was in the ocean water during the Permian-Triassic extinction, the event that eliminated approximately 96% of all marine species? The answer required molecular paleontology, extracting and identifying chemical compounds from boundary-age rock. What they found in Permian-Triassic boundary sediments from China, Italy, and Austria was isoranierateinae, a hydrocarbon pigment produced exclusively by chlorobiaceae, the family of green sulfur bacteria that perform photosynthesis only in sulfide-rich anoxic conditions. Thanks to their presence in the photic zone is unambiguous evidence. The toxic, oxygen-depleted chemistry had reached the sunlit surface layer. Jonathan Payne of Stanford University and Lee Kump of Penn State published complementary analyses in 2004 and 2007 using carbon and boron isotopes to reconstruct the rate and magnitude of simultaneous ocean acidification. Paul Wignall of the University of Leeds documented the spatial extent of anoxia using iron speciation data in a 2003 Science paper. Anoxic conditions extended into shallow shelf environments within tens of thousands of years of the initial Siberian traps volcanism. The recovery from this event took approximately 10 million years, the longest biotic recovery of any mass extinction in the fossil record. The Permian-Triassic kill chain operated through 5 overlapping mechanisms. In volcanic CO2 caused warming. Warming reduced oxygen solubility. Anoxia developed in the deep ocean and propagated upward. Sulfate-reducing bacteria colonized the anoxic water column, producing hydrogen sulfide. H2S vented into the atmosphere, destroying ozone. Without ozone, UV radiation reached the surface. Spore and pollen assemblages from the boundary interval show widespread UV damage mutations in plant DNA, documented by Vischer and colleagues in 2004 in PNAS. Each mechanism amplified the others. No single cause can be isolated. The system was pushed past 5 thresholds simultaneously, and each threshold crossing accelerated the next. Those 5 thresholds: ocean pH, oxygen saturation, sulfide concentration, stratification depth, and UV flux, have been measured in the geological record. The distances between modern ocean baselines and those thresholds are known. They are not equal. Some are further away than others, but they are all moving in the same direction. And they have been moving in that direction continuously since industrialization began. The mainstream interpretation holds that the Permian-Triassic extinction required the Siberian Trap's extraordinary eruption volume, estimated at 3 million cubic kilometers of lava, to initiate a kill chain that no single threshold could have triggered alone. A minority argument, increasingly visible in the geochemical literature, is that the modern ocean is currently approaching all 5 threshold conditions simultaneously. Rising CO2, falling pH, expanding anoxia, increasing stratification, and measurable ozone thinning. The Permian-Triassic event required all 5. All 5 are currently trending in the same direction. A 2022 paper in Science by Justin Penn and Curtis Deutsch at the University of Washington used biophysical models calibrated against the Permian-Triassic record to project future ocean oxygen and temperature conditions. Their conclusion: under high-emission scenarios, by 2300, tropical and mid-latitude ocean conditions could approach Permian-Triassic analog states for 40 to 50% of marine species, not identical to the N-Permian, analogous to it. The distinction matters. Both matter. Number 9, the stagnant ocean. When ocean circulation fails, the water column becomes a permanent graveyard. The Proterozoic ocean did not circulate meaningfully. Without significant temperature gradients between equatorial surface water and polar deep water, both warm and both oxygen depleted, the thermohaline circulation that today moves oxygenated surface water into the deep ocean effectively ceased. The ocean was a planetary standing pool, no vertical mixing, no renewal, just slow accumulation of the metabolic waste products of a billion years of microbial activity. The conditions that Canfield described: the Atlantic Meridional Overturning Circulation, AMOC, is the dominant mechanism by which oxygenated surface water reaches the deep Atlantic today. It also modulates northern European climate by transporting tropical heat northward. Stefan Rahmstorf of the Potsdam Institute for Climate Impact Research published a 2015 paper in Nature Climate Change documenting a measurable slowdown in AMOC over the 20th century, using sea surface temperature patterns as a proxy. A 2021 paper in Nature Geoscience by Nicholas Boers, also at the Potsdam Institute, analyzed 8 independent AMOC indicators and identified critical slowing down, a statistical signature that appears in dynamical systems as they approach a tipping point. The mechanism is straightforward. Thermohaline circulation depends on the density difference between warm, less dense surface water and cold, dense deep water. As surface temperatures rise and polar ice melts, freshwater input from Greenland and Arctic glaciers dilutes North Atlantic surface water, reducing its density. A fresher, warmer surface mass does not drive deep circulation. The deep ocean stops receiving oxygenated water from above. Oxygen minimum zones expand upward. The geological consequence, visible in every Mesozoic oceanic anoxic event, is progressive deoxygenation of the deep ocean. As circulation fails, existing dissolved oxygen is consumed by decomposition bacteria and never replenished. The process, once initiated beyond a threshold, is self-sustaining. Warm, stratified, low circulation oceans do not spontaneously reoxygenate. They require large-scale cooling, effectively glaciation, to re-establish the density gradients that drive circulation. The Mediterranean Sea, enclosed and poorly ventilated, periodically generates sapropples, dark organic-rich sediment layers chemically identical to OAE deposits, during periods of reduced deep-water formation. The most recent Mediterranean saproppal event, approximately 9,000 years ago, coincided with freshwater input from melting glaciers into the North Atlantic. They thought that same forcing is now active again in the same ocean system. The IPCC Sixth Assessment Report characterized a complete AMOC collapse before 2100 as unlikely but not impossible under high-emission scenarios. Boers and colleagues do not predict collapse on a specific timeline, but identify the statistical fingerprint as consistent with an approach to a critical transition. The mainstream and the cautious minority are debating the timeline, not the direction. The most disturbing reading in the oceanographic literature is not that circulation will collapse suddenly, it is that partial sustained weakening, already documented, produces progressive deep ocean deoxygenation without any single dramatic event. No visible threshold moment, just slow, a steady accumulation of the conditions that preceded every black shale deposit in the fossil record. Number 10. The UV catastrophe. 3 times in Earth's history, the ozone layer was chemically destroyed by processes originating in the ocean. The mechanisms are currently running. The connection between ocean chemistry and sky chemistry is not intuitively obvious. But the Permian-Triassic boundary, the late Ordovician extinction, and the Toarcian event share a specific biological signal preserved in the rock record: mutant plant spores. UV radiation at the wavelength blocked by the ozone layer, 280 to 315 nanometers, causes specific identifiable damage to plant DNA. Those damage signatures appear in the fossil record at extinction boundaries. The sky broke because the ocean changed. Jens Henk Vischer of Utrecht University and colleagues published the discovery of UV-damaged spores from the Permian-Triassic boundary in PNAS in 2004. The mutagenic signature, specifically the formation of UVB-induced cyclobutane pyrimidine dimers in fossil spores, was identified in boundary-age material from China, Austria, and Greenland. 3 geographic locations, the same damage, the same moment. The evidence is not regional, it is global. The mechanism connecting ocean euxinia to ozone depletion runs through hydrogen sulfide. Lee Kump, in a 2005 Nature paper, modeled the atmospheric consequences of large-scale H2S venting from a euxinic ocean. At flux rates consistent with end-Permian euxinia, hydrogen sulfide and its atmospheric derivatives could reduce stratospheric ozone by 40 to 70% within decades. ESA's second mechanism for rapid ozone destruction was proposed by Adrian Mellott of the University of Kansas and Brian Thomas of Washburn University in a 2004 Astrophysical Journal Letters paper. Ionizing radiation from an Ordovician gamma-ray burst photolized nitrogen and oxygen bonds in the upper atmosphere, producing nitrogen dioxide and nitric acid at concentrations sufficient to destroy ozone globally. 3 independent routes lead to the same endpoint: ozone collapse and UV flooding of the surface ocean. First, ocean euxinia produces H2S, which vents to the stratosphere and catalytically destroys ozone. Second, large igneous province eruptions release halogens, chlorine and bromine, which react with ozone in the same catalytic cycle exploited by industrial CFCs. Or third, energetic particle events photolize stratospheric molecules directly. All 3 have geological records. All 3 produce the same biological fingerprint: UV-damaged spores, selective extinction of surface-dwelling organisms with soft tissue, and survival bias for deep-water species shielded by the water column itself. When ozone fails, water is the only remaining UV shield on the planet. Anything living in the top 10 meters of the ocean becomes exposed to sterilizing radiation. The mainstream interpretation holds that modern stratospheric ozone is recovering from CFC-related depletion following the Montreal Protocol, and that hydrogen sulfide flux from the modern ocean is insufficient to replicate end-Permian ozone damage. Some researchers, building on Kump's 2005 model, might argue that the relevant threshold is not global H2S flux, but localized venting events, that a large-scale shelf eruption in a semi-enclosed basin could produce temporary but severe regional ozone, thinning over areas of dense marine productivity. The most speculative synthesis integrates all 3 mechanisms as potentially concurrent: a sufficiently destabilized ocean chemistry initiating simultaneous H2 S venting, and volcanic halogen release, producing a compound ozone attack with no modern counterpart, but a clear N Permian precedent. The ozone layer over Antarctica still thins measurably every austral spring. Volcanic eruptions release halogen compounds that transiently affect stratospheric chemistry. These are not threats in isolation. They are demonstrations that the atmosphere's chemical stability operates on narrow margins. And that the ocean, through continuous gas exchange, is the primary regulator of those margins. When ocean chemistry shifts towards sulfide production, the sky follows. The geological record says so, unambiguously, repeatedly. The deep pattern, convergent chemistry across 1 billion years. Across all 10 entries, the same set of chemical transitions appear and reappear. Oxygen loss, sulfide accumulation, pH collapse, stratification, ozone thinning. These are not 10 separate threats. They are 5 phases of a single cascade, and every entry on this list represents a different entry point into that cascade. The cascade works as follows. Carbon loading, from volcanism, from biological weathering, from methane clathrate release, now from industrial combustion, raises atmospheric CO2, temperature rises. And warm water holds less dissolved oxygen. Oxygen minimum zones expand. With less oxygen available for aerobic decomposition, organic matter accumulates on the seafloor. Sulfate-reducing bacteria proliferate. They produce hydrogen sulfide. Euxinic conditions develop in the deep and propagate upward. H2, S vents into the atmosphere. Ozone thins. UV penetrates the surface ocean. Photosynthetic plankton, the foundation of ocean oxygen production, are exposed to lethal radiation. Oxygen production drops further. The cascade tightens on itself. This is not a chain of probabilities. Each step has been documented in the geological record independently. The N Permian ran all 5 phases simultaneously. The torsion ran 3. The Devonian ran 2. The PETM ran 1 and triggered partial others. The question is not whether the steps are mechanistically connected. They demonstrably are. The question is how far along the chain the current trajectory will travel, and at what rate. Several modern indicators already sit within measurable distance of geological thresholds. Ocean pH has dropped 0.1 units since industrialization. The torsion event involved a 0.3 to 0.4 unit drop. The gap is a factor of 3. Global ocean oxygen content has fallen approximately 2% since the 1960s. Oxygen minimum zones have more than doubled in extent since 1950. AMOC circulation indicators show statistical slowing down signatures. Arctic shelf methane seep fields are expanding. Ancient viral and microbial material is being released from thawing permafrost at rates without Holocene precedent. And none of these individually constitutes a mass extinction event. Collectively, they constitute a direction. And direction in the geological record is what matters. The fossil record does not, in most extinctions, record gradual linear deterioration. It records thresholds, intervals of apparently stable chemistry followed by transitions. The Permian-Triassic transition from onset to peak extinction lasted approximately 60,000 years. The torsion carbon injection occurred in under 35,000 years. The PTM thermal maximum developed within 20,000 years. These are geological eye blinks. They are still longer than the entirety of recorded human history. There is a structural irony embedded in every entry on this list. The organisms that detected early-stage chemical deterioration were the same organisms responsible for buffering it. Plankton that fix carbon and produce oxygen, filter feeders that absorb excess nutrients, reef systems that sequester carbonate. When they fail under chemical stress, the buffering capacity disappears precisely when it is most needed. The system loses its governors as it accelerates. Donald Canfield's 1998 paper identified the Proterozoic ocean not as a primitive phase Earth outgrew, but as a thermodynamic attractor. A stable low-energy state the ocean gravitates toward when the oxygen budget falls below a critical level. Euxinia is not unusual in Earth's history. Oxygenated, circulating oceans are the exception. Complex life evolved inside and depends entirely upon the exception. The chemistry documented in these 10 entries is not ancient and inert. It is present, measurable, and directional. Isoranierotane, the molecular pigment that proves photic zone Euxenia reached the surface during the Great Dying, is already forming in modern restricted basins. Pteropod shells are already dissolving in Southern Ocean water, with pH chemistry approaching torsion analogs. Methane seep fields are already expanding on Arctic continental shelves. Oxygen minimum zones are already growing in the same ocean basins that produced O-A-E black shales 94 million years ago. Ancient viruses revived in 2022 had been frozen since before modern humans existed. The geological record does not predict, it demonstrates. It shows what these mechanisms produced the last time each threshold was crossed. The thresholds have not moved, the chemistry has not changed. The periodic table is the same one it has always been, for the first time in 4.5 billion years. And if one species can measure all of these processes in real time, simultaneously, with instruments distributed across every ocean basin on the planet, that is not reassurance. It is the only thing that distinguishes this moment from every previous occasion when the cascade began. The question the record raises is not whether the chemistry is capable of these transitions. It ran all of them, repeatedly, across 600 million years of documented ocean history. The question is what, if anything, will be different this time. The record does not answer that question. It only makes it impossible to claim we were not warned.